Hello, everyone, and it's an honor for me to present Professor Gepstein as the speaker for the talk today about innovative biological solutions for the study and treatment of cardiovascular disorders. Professor Gepstein graduated his MD and PhD studies at the Rappaport Faculty of Medicine of the Technion Israel Institute of Technology. During this period, he was involved in the development of an electroanatomical mapping techniques CARTO system, which became the state-of-the-art technology for the treatment of complex cardiac arrhythmias. Dr. Gepstein completed his residency in internal medicine and cardiology fellowship at Rambam Healthcare Campus and fellowship in cardiac electrophysiology at the University of California, San Francisco. Currently, he serves as the director of cardiology at Rambam Healthcare Campus and holds the position of professor of physiology and medicine cardiology and the Sohnis chair in tissue engineering and regenerative medicine at the Technion's Faculty of Medicine. Professor Gepstein was recently appointed as the director of the research division at Rambam. Professor Gepstein's research activities focus on the areas of cardiac electrophysiology, stem cell biology, studying of inherited cardiac disorders, and the establishment of novel gene and cell-based strategies for the treatment of different cardiac disorders. Professor Gepstein, the stage is yours.
Thank you very much, Owen. I'll try to. Could you stop?
I would just like to mention that there are Q&As that people can ask during the conversation.
Okay, so I shared my screen. I hope you can all see it.
Yeah.
Okay, so good afternoon, everybody. Thank you very much for the kind words and for the invitation to share with you my thoughts about using biological solution for the study and treatment of cardiovascular disorders. One of the, I think, major problems we have as translational scientists is the big difference that we have between physicians, what they know today, and the large advancements that we have in basic scientific methods that is actually incredible the way it's developing over the years. And us as scientists that are trying to bridge the gap between basic research and translational research, I think one of our jobs is to try to nurture these methodologies and to use them for our research. And Nature Methods has a contest every year of the method of the year. And what I'll try to do is to share with you some of these methods that was introduced over the last 2 decades, and how we have been using them in the lab to help us study and potentially develop treatment modalities for different heart disorders. And the first technology is, I would like to share with you, is actually quite old already, is the human embryonic stem cell technology, in which human blastocysts generated for in vitro fertilizations were used to isolate pluripotent stem cells from the inner cell mass. These stem cells, which were called human embryonic stem cells, were able to be propagated indefinitely in the culture dish in the lab, and then be coaxed to differentiate into every cell type in the body. And we had the opportunity to use them early on. And back in 2001, we were able to establish an essentially spontaneous differentiation system of these human embryonic stem cells to derive the first beating human heart cells in the dish. And we then use them for a variety of reasons. And of course, the most obvious application is maybe use them to regenerate the heart, to repair a damaged heart that has undergone a myocardial infarction for the treatment of heart failure. And in order to do this, it's not enough that we generate heart cells. We have to show that these cells can integrate and beat in synchrony with host tissue. So we did several studies over the years. In this specific study, we took our human heart cells, which you can see here, and plated them next to rat heart cells. And within the few hours, the two tissue types started to beat in synchrony. So when the human heart cells beat, also the rat heart cells beat. And this is true integration because we did this study on an electrical mapping system that shows that there is indeed electrical propagation propagating from the human tissue to the rat tissue and so on. We then continued to demonstrate this electrical integration also on the in vivo level. And finally, we wanted to show whether this can actually be used to improve the myocardial performance of a damaged heart. To this end, we used a rodent model of a heart attack in which we ligated, occluded by surgery, the left anterior descending coronary artery of the rat heart, created a myocardial infarction in which the area supplied by this artery dies and is replaced by scar tissue. A week later, we came back and injected our human heart cells into the infarcted territory in the rat heart, and then followed them by echocardiography, functional. And as you can see, a normal heart function is about, fractional shortening is about 35%. When we create an infarct, it drops to about 20%. If you don't do anything, just inject cell line, you can see that the function of the heart continues to deteriorate. This is what we call adverse remodeling, and we see it in patients after a myocardial infarction. If we transplant cells that are not heart cells, you can see that this remodeling still exists, negative remodeling. But if we inject our human heart cells into the infarcted area, you can see that not only that we prevent this remodeling, there's even some improvement in the function of the rat heart. So this was an initial proof of concept that we can actually improve the function of a diseased heart by transplantation of human heart cells. But we did notice several shortcomings at that time and things that need to be overcome in order to take this to the clinic. The first is getting enough heart cells. Our differentiating system was not very efficient. Only a few % of the cells became heart cells, and we could not generate the billions of cells required to regenerate a human heart. So over the years, we and several other labs used lessons from development biology, and now the field has many protocols in which one can use human pluripotent stem cells and differentiate them into almost 100% purified population of heart cells, as you can see here. And now we can make about 1 billion heart cells for a cost of about $1,000, something like this. So generating enough heart cells is probably a problem that can be solved. The next issue is, what do we do with immune rejection? Since although these cells are embryonic, they are not as immunogenic in as adult organ, they are still expected to be rejected because we are not dealing with an autologous transplantation. So there are many solutions to this, potentially using immune suppression because these cells are less immunogenic, creating universal cell lines in which we modify them genetically to be less immunogenic. But maybe the ultimate solution may be the technology introduced by Shinya Yamanaka, the induced pluripotent stem cell technology, work from which he got the Nobel Prize for, in which one can take a patient-specific adult cell, like a skin cell or a blood cells, then by transiently introducing the expression of 4 transcription factors, one could regenerate these adult human cells to generate cells that resemble embryonic stem cells, which Yamanaka called iPS cells, or induced pluripotent stem cells, which can then become any cell type in the body. You can coax the differentiation to liver cells, heart cells, nerve cells, and so on. And since they are derived from the specific patients, if you transplant them to this patient, they will not be rejected. Similarly, since their genetic composition is identical to these patients, they could generate, in essence, a patient in a dish model, and we use them to study different genetic disorders, as well as to study drug development and drug screening studies. And this turned out to be a robust technology. This is one of our earliest work in which we took cells from patients, reprogrammed them to make iPS cells, and then you use your favorite differentiation system to generate the patient-specific heart cells that are beating in a dish. Okay, so we talked about now we can make not only human heart cells for the first time, but even patient-specific heart cells. The question is, what heart cells are we generating? And it turns out that we are generating a mixture of different heart cell types. Most of the cells have a ventricle-like phenotype, the lower chamber of the heart. Some of the cells have an atrial-like phenotype, resembling the upper chamber of the heart cells, and some resemble SA node cells or the pacemaker cells of the heart. But the heart has also additional cell types. We have different conduction system cells, and we have even non-muscle cells, such as epicardial cells, endocardial cells, vascular cells, fibrous, and so on. So the next challenge would be not only to generate heart cells from patient-specific iPS cells, but to generate different specific heart cell types. And over the years, together with our collaborators, Gordon Keller in Toronto, Canada, we've been able to develop protocols to allow us to generate almost any cell type in the heart. In these specific studies, we were able to generate cells that are either have a ventricular-like phenotype or an SA node pacemaker-like phenotype. What can you do with ventricular cells? The ventricular cells, you want them to repair the myocardial infarction to treatment of heart failure. So you can use them for transplantation studies in the ventricle to repair the damaged heart. What can you do with SA node pacemaker-like cells? Perhaps you can use them as an alternative to electronic pacemakers. You know that when we age and our conduction system deteriorates, the heart may beat too slow because of problems in the pacemaker cells or in the conduction system. And this requires the implantation of an electronic pacemaker. But what if instead of implanting an external device, what if we just transplant healthy pacemaker cells and therefore restore the normal pacemaker function? So to this end, we studied the ability of SA node pacemaker cells that we generate to pace the heart. We started with an in vitro study in which we took a ball of SA node cells that you can see here, put them on a ventricular tissue, and as you can see, the SA node cells started to drive the electrical activity of the heart. They behaved like a general and native pacemaker. In contrast, if you put ventricular cardiomyocytes, as you can see here, they do not pace the tissue, but rather become integrated into the existing tissue. To demonstrate that this can also work in vivo, we transplanted our SA node cells into the apex of the left ventricle of the rat heart. And 2 weeks later, we took out the heart and studied it with an optical mapping system that allows us to study how the electrical activity propagates in the heart. Note that the electrical activity starts from red, then to yellow, green, light blue, and so on. So the pacemaking activity in the ventricle actually starts in the septum in the rat, and not where we transplanted the human heart cells in the apex. So this a little bit frustrated us because it means that the SA node cells were not pacing the heart. But it's actually not surprising because in the heart, there is a rule that the fastest pacemaker dominates. And since the rat heart beats faster than the human cells, about 300 beats per minute, then the rat conduction system dominates, and that's why our cells are not pacing it, as you would expect by the law of the fastest pacemaker dominates. So to slow down the rate, we generated a model of slow heart rate, a scenario in which we implant electronic pacemakers in patients. We generate what we call complete AV block, dissociating the electrical activity between the atria and the ventricle pharmacology. And after slowing down the rate of the rat heart, as you can see, our pacemaker cells started to pace the heart. Now the red area is exactly where we transplanted the cells. So like a good pacemaker, when the heart rate is fast, it doesn't kick in. But when the heart rate becomes very slow, it kicks in and starts to pace the heart. When we stop the drugs and the heart restores its normal activity, you can see that our cells stop pacing. In contrast, if you put ventricular cells in the same site, you can see that when we create AV block, a slow heart rate, our ventricular cells do not pace the heart. They remain passive. And you see that there is either no activity or very slow activity from an escape rhythm. So this actually is important because we do not want ventricular cells to behave as pacemaker cells because they will create an arrhythmia. But if you transplant the correct cells, the SA node cells, then they could pace the heart. And maybe in the future, we can use them as an alternative to electronic pacemakers. Okay, the next challenge is, after showing that we can make ventricular cells that can be used for infarct repair, is how do we assure that these cells survive well after transplantation and that they generate an adult structure, an organized structure, and actually improve the function of the heart. So to this end, we're moving more and more, not only to transplanting individual cells into the damaged area, but actually generating an engineered tissue through the concept of tissue engineering. The idea behind tissue engineering, that our organs are not made only from cells, they are made of cells that grow on top of a scaffold made out of extracellular matrix. These ECM proteins allow to give the organ the shape and support the growing cells. So tissue engineering combines cells with polymers, either synthetic or biological, in order to generate tissue substitutes or even organs. And over the years, we've been using a number of tissue engineering concepts with a number of collaborations, either using an hydrogel approach or a decellularization, recellularization approach, or more recently also 3D bioprinting. And I'll give you some example. This is our initial hydrogel approach, what we call the engineered heart tissue, in which we collaborated initially with the group of Thomas Heschenhagen and Wolfram Zimmermann in Germany. This is their work initially using rat heart cells, in which they use rat together with collagen to make a mixture. They put it in a mold to generate rings. And then after the rings are made, they put it on a stretcher device. This is like working out for the cells, similar like when we work out our muscle hypertrophies, also the cardiac cells hypertrophy, and we can generate a tissue that starts to beat, and we can have this like rubber band like beating structures that can generate a significant amount of force. And now we can do this both with our human embryonic stem cells as well as with our human iPS cells, and generate these spontaneously beating structures which look like beating calamari, we call them in the lab. So we can make not only cells, but even 2D and 3D structures, which can then be used for a variety of applications, as I will discuss later. An alternative approach is to go further on and not only generate a three-dimensional tissue in general, but to try to use a strategy that will also allow us to retain the anatomy, the desired anatomy of the tissue. And to this end, we've been using an organ decellularization, recellularization approach, in which one can take, for example, a rat heart or a pig heart, use enzymatic dissociation to dissociate all the cells from the organ and remain only with the extracellular matrix. One can then reseed the desired cells, in our case, human cells, and generate a tissue with the appropriate microscopic and macroscopic structure. So in this case, for example, we've been using a ventricular tissue scaffold that we generate from this decellularization, recellularization process. We recellularize it with the human ventricular cells, and we generated and beating engineered ventricular patch, as you can see here. So now we can use this patch, which retains the microscopic structure of the tissue, and use it, for example, for transplantation studies on the infarct area in the scar. So you can combine cells with the anatomy to generate now a more clinically relevant structure of the tissue. Both what we described before, the hydrogel and the decellularization, recellularization, result in generation of a patch, that you will need to transplant it during surgery in order to repair the heart. But I am a cardiologist, and as you know, cardiologists don't like to give work to surgeons. So we are trying also to develop an alternative strategies in which we can maybe generate the engineered tissue inside the infarct. The idea is to transplant the polymers together to the cells into the infarcted area, and the polymer will serve as two purposes. First, it will strengthen the scar and maybe prevent the abnormal remodeling that I've described before after myocardial infarction. And second, it will allow entrapment of the cells and provide a supportive environment for the cells for a better graft. So to test this, we've been collaborating with Dror Selectar from biomedical engineering in the Technion, in which he developed a PEG fibrinogen polymer, which can come in a liquid state and then can be gelatized using different approaches. Here we use UV to perform gelatization, and you can see this is the structure that is formed. We can now inject a cell line or heart cells with the polymer in the heart and follow what happens. And as you can see, we can see a drastic effect. This is the control heart. Note how thin the scar becomes eventually and the dilatation of the ventricle, whereas the hearts that were injected with the polymer and the cells can generate significant improvement in the structure and function of the heart. Note the anterior wall is much more thickness with a lot of viable cells within it. And if we combine the four groups, cell line, polymer alone, cardiomyocytes, and cardiomyocytes and polymer, we can see that the optimal results is transplanting cells with the polymer. Okay, well, what is the status now of this field? So we're really becoming close to clinical studies and work in the from our collaborators, as well as some of our competitors, demonstrated the ability to engraft and regenerate a significant portion of the infarct in already primate hearts, as you can see here, in such a way that we can regenerate almost 80% of the infarct. So the fact that we can make enough cells, the appropriate cells, and that you can generate significant grafts in large animals brings us very close to an eventual clinical study, which we hope will start in the next few years. So I've talked about the use of induced pluripotent stem cells for regenerative medicine. What else can we do with them? So one interesting application is to use them for the study of genetic disorder. The idea is if a patient has a genetic disorder affecting his heart, for example, a cardiomyopathy affecting the muscle, or what you call a channelopathy affecting the electrical activity, one cannot really study the patient disease because you cannot take out his heart. He needs it. But if one takes a patient-specific skin cells, reprograms them to generate iPS cells, and then coax their differentiation in the dish to heart cells, these heart cells should have exactly the same genotype of the patient, and therefore should have exactly the same phenotype and can be used to model a genetic disorder. To demonstrate the feasibility of this concept, we studied a genetic disorder called long QT syndrome a few years ago, in which we had a young patient coming out, admitted to our hospital after she had cardiac arrest in the street. When she came in the hospital, she had these repeated episodes of polymorphic ventricular tachycardia that required multiple electrical shocks. We learned that the patient had a family with several members dying at a young age. And we showed that she has an abnormal ECG with a long QT interval. This is why it's called the long QT syndrome. So we screened the family and identified a mutation in the KCNH2 gene, which encodes a potassium channel. So the disease involves a reduced potassium current, which results in a delayed repolarization. And that's why you see the prolonged QT interval, which results in this arrhythmia. So we then wanted to see whether we can generate an iPS model of this disease. So we took some skin cells from the patient, made using Yamanaka technique, induced pluripotent stem cells of that specific patient, and then drove their differentiation into generation of human heart cells. At that time, we still didn't know how to make specific heart cell type, so we got a mixture of the ventricular cells, atrial cells, and nodal cells. This is from a healthy control individual in which we generated iPS cells. When we compare these cells, heart cells that we generated from our patients, you don't have to be a cardiac electrophysiologist to see that the action potential is much significantly more prolonged in the patient's cells. That's because of the mutation in the potassium channel that delays the repolarization, that delays the return to normal activity. Not only that, but her cells, some of her cells also developed arrhythmias, as you can see here. So you had a patient coming with an arrhythmia, you took some skin cells, made heart cells, and the patient-specific cells in the dish had the same arrhythmia as the patient. So now you can study the mechanism of the disease in a dish, and also study potential drugs that may be beneficial for that specific patient. Here, for example, we tested a drug that opens an alternative potassium channel called pinacidil, which shortened the action potential duration and prevented the arrhythmias in a dish. To see whether this concept can actually guide us in clinical decisions, we studied another genetic disorder in a Bedouin family that also displays sudden cardiac death in a young age. It's called CPVT, catecholinergic polymorphic ventricular tachycardia. This disease affects calcium handling in the sarcoplasmatic reticulum. And when these young kids exercise, they can develop this potentially lethal arrhythmia. So in some of these patients, we implanted the defibrillator, which saves their life. Every time they have this arrhythmia, they get a shock, but the shock is painful and releases adrenaline to the bloodstream. So every time they get an arrhythmia, they get a shock, they develop another arrhythmia, another shock. So sometimes they come to the hospital with 30 sequential shocks, which is not very nice. So we wanted to see if we could help them. So to this end, we generated the iPS cells from these patients, generated their specific heart cells, and then studied their calcium activity, since this disease affects the calcium handling within the heart cells. Note that this is the normal calcium transit in a healthy control iPS-derived heart cells, whereas heart cells derived from iPS from our patients have significant arrhythmogenic activities and abnormalities, as you can see here. So it's not surprising that they have this really arrhythmic phenotype. We then wanted to see whether we can test potential drugs that may be beneficial from that patient. So we tested a variety of drugs that are FDA approved for other indications. And we noted that some of them can worsen the electrical activity, the abnormalities, for example, something that works like adrenaline, whereas there were a few drugs that resulted in reduction in the arrhythmogenic activity. Note, for example, this treatment, flecainide, which is an anti-arrhythmic drug used for other type of indications, but it had a significant anti-arrhythmic effect on the patient-specific cells. So we then went back to the patient and put him on a treadmill. So as you can see, when it starts to exercise just a little bit, it starts to have this ectopic electrical activity arrhythmias. Whereas if we treat him with flecainide before they working out, it completely eliminates the generation of arrhythmia. So now the patient is receiving flecainide as a potential treatment for him. So we are really continuing this concept of disease in a dish for studying arrhythmias and other cardiac disorders, as well as for developing newer treatment for these patients. And the next stage involves, again, looking at specific heart cell types. We told you about looking at ventricular cells and pacemaker cells. In another project, we are generating atrial cells. In this case, by manipulating the retinoic acid pathway, this allows us to generate purified population of atrial cells, the upper chamber of the heart. And these cells have the appropriate atrial cell properties. These are their electrical activity, in which we show that they have atrial-specific currents that are not present in the ventricular cells. What can we use atrial cells? We can use them to study the most common cardiac arrhythmia called atrial fibrillation, which is the major cause of stroke in the world. And you can see that in order to generate atrial fibrillation, it's not enough to study the cells at the cellular level as we did for the CPVT and the long QT. Because atrial fibrillation is what we call reentrant arrhythmia. And to model such an arrhythmia, you need a tissue. So we're moving from the single cell to the tissue level. Not only that we can make atrial cells, but we can also make an atrial tissue. We're starting with a 2D tissue, as you can see here. And you can see that by plating a few million cells, we can make like a 2D small atria in a dish. And you can see the beating in this tissue. And then we can study them using an optical mapping system. We load the cells with voltage-sensitive dyes. And then by following the fluorescence, we can actually study how the electrical activity propagates in the tissue. And then you can study how the basic electrical physiological properties of the tissues, and you can also induce arrhythmias. For example, here we are generating rotors, which are thought to underlie fibrillation. You can see it looks a little bit like a hurricane. I hope you can see it. Sometimes it doesn't play well over Zoom. And then we can use this tissue with the mapping system that we developed to study electrical activity at baseline, to learn how this electrical activity starts, as you can see here, how it becomes sustained, and how it can break to many wavelets simulating fibrillation. So now we have a fibrillation model in a dish to study such arrhythmias. Our next stage is to take it even further and introduce the 3D anatomy in our models. To this end, we are starting to use 3D printing, and which we are printing not only materials, but also cells. This is work in collaboration with Tal Dvir, in which we use our human atrial cells, take CT scan from patients, and then generate an atrial tissue with the appropriate anatomy, the pulmonary veins, and so on. So we can generate an atrial tissue and can generate an atrial fibrillation model that also involves the anatomy of the tissue in our model. So we talked about moving in our modeling from single cells to 2D to 3D. What else can we introduce? We can also introduce another new biological approach, which is gene editing using CRISPR. And in order to show the ability to use CRISPR, both for our modeling study as a potential therapy, we studied another inherited disorder called the short QT syndrome. Remember that I showed you that the long QT, when the action potential is too long, is bad. When you had loss of function of potassium currents, we had a prolonged action potential duration, and this results in arrhythmia. But also, if you have interestingly a too short of an action potential duration, this can also cause an arrhythmia. And this is usually due to gain of function mutation in potassium channel, too fast repolarization. So to model that, we study a patient family that from Germany. We got the cells from these German researchers, generated an IPS line, differentiated them into heart cells, and then studied their action potential. You can see that here, this is the healthy control, whereas in this case, in contrast to the long QT syndrome, the patient-specific heart cells had a shorter action potential duration relative to the healthy control cell. And in order to show that it's indeed the specific change in the DNA in the KCNH2 gene that was identified is responsible for this phenotype, we use gene editing, CRISPR, to correct this mutation with the wild type variant. And we then studied the corrected cells for their action potential properties, Note that the short QT cells have a short action potential duration relative to the healthy control that we studied before, but they are also shorter relative to the corrected cells in which they have exactly the same genetic background as the short QT cells. The only difference is that the mutation was corrected, and this proves that indeed this specific change was responsible for the phenotype. We then demonstrated the ability of this model to model arrhythmia at the tissue level. So you can see that the short QT syndrome cells had 80% of the tissues generating arrhythmias, whereas the healthy control, only 35% of the cells developed the tissue developed arrhythmias, whereas the corrected cells had no arrhythmias developed. So we can use CRISPR both as the best control for our models, as a way to study the potential functional significance of what we call a VUS, variants of unknown significance, if you have a genetic change in which you are not sure whether it causes a genetic disorder, and also potentially as a treatment for genetic disorder. We also use this model to try to find an optimal solution for this patient. So the most obvious solution was try to find a drug that blocks the potassium channel. Because if you remember, I told you that the problem is too much activity of a potassium channel. So if you take a drug that blocks this potassium channel, it should help the phenotype. But interestingly, when we used a drug, sotalol, that blocks the potassium channel, usually it didn't have no effect. We still had the same action potential duration, the same percentage of inducing arrhythmia. We think is that sotalol interacts with the channel exactly at the site of the mutation, and the mutation prevents appropriate binding of sotalol to the channel. So we studied an alternative drug, quinidine, which blocks several channels, not only one channel, and we saw that quinidine prolongs the action potential duration and prevents arrhythmia in our cells, and indeed quinidine is the optimal therapy in this patient population. And the last technology I would like to share with you does not necessarily involve pluripotent stem cells, but rather a different technique called optogenetics. Optogenetics started when scientists realized that algae move away or toward light because of the presence of light-sensitive ion channels in their cells, with the prototype channel called channel rhodopsin 2, which is a cationic specific light-sensitive channel. So when you shine blue light on this channel, it opens and allows positive ions to enter the cell. So scientists then cloned this channel and expressed it in neuronal tissue, either in cells or in animals such as rat and primates. And they developed an entire new field in which one can use light to allow specific perturbations to neuronal network. So now people use this to study the brain, and this really transformed neuroscience. And we wanted to see whether we can take this approach also to the cardiac field and try to control the electrical activity of the heart with light. So we used adeno-associated viral gene delivery to deliver the transgene encoding channel rhodopsin 2 to the apex of the right ventricle, to the apex of the ventricle of the rat heart. 2 weeks later, we took out the rat heart, and it can beat outside the body. So you can see it beating happily. We will now shine flashes of blue light, and I hope you can see that every flash of blue light paces the heart. The blue light activates the channel rhodopsin, generates depolarization by sodium channel and action potential, and we actually generate a light-induced pacemaker, in which we can pace the heart at different rates, similar to electronic pacemaker, but without contact. We can also do it when the heart is still in the chest. I hope you can see that the heart is beating. We shine flashes of blue light, and the heart is paced by light. So we can control the heart by light. Maybe we can use a light-induced pacemaker instead of an electronic pacemaker. But can we do more? And here, for example, we wanted to show that we can pace the heart from more than one side. We know that patients that have abnormal electrical activity can develop what we call desynchronization. This is when the heart not only that doesn't contract well, but also contract in a desynchronized manner, that one area contracts before the other. In order to try to solve this, we do what we call the cardiac resynchronization therapy, in which we pace the ventricle from two different sides simultaneously to try to synchronize its activity. This is a few $1 billion market a year, but not all patient responds, probably because we are limited to the number of sites we are pacing the heart from, and where we can pace it from, where the veins allow us to go to place the electrodes. So we wanted to see whether we can do an alternative optogenetic-based resynchronization therapy, because you're not limited from how many sites you can pace the heart from with light. And in this case, we deliver channel rhodopsin into 3 or 4 different sites in the left ventricle of the heart. We then looked at how the electrical activity heart looks during normal rhythm. So you see normal rhythm is very synchronous. We then simulated the synchrony by pacing the right ventricle. So this is when the heart is desynchronous. Look, it takes more time to activate it electrically. But when we shine light on the 3 sites that we deliver the channel rhodopsin, we can restore the synchronous activity of the heart. So if we quantify this, normal activity of the heart is very synchronous. It takes only 15 milliseconds to activate the entire ventricle. If one has desynchrony, we simulate this by pacing the right ventricle. You can see that time goes up to 40 milliseconds. So the heart doesn't contract efficiently because it's not synchronized. But if we optogenetically pace it by light from several different sites from the ventricle, we can restore the normal synchronic contraction of the heart. And lastly, can we do the opposite? Can we not only generate electrical activity, but also stop electrical activity? Maybe we can use it as a new approach to treat ventricular fibrillation, like the defibrillator I described, or atrial fibrillation that I've described you about. The problem when you give electrical shock to treat this disorder, that it's extremely painful. But if we use optogenetics, it will not affect the nerve endings of the heart, only the heart cells, and therefore we can maybe have a painless defibrillation. So to test this, we wanted to use an optogenetic strategy to stop electrical activity. So we are using 2 approaches. Either we are using a chloride-based light-sensitive channel, which induces hyperpolarization, negative membrane potential, and this prevents the initiation of new beats. Or we can use the same channel rhodopsin that if you shine light for a short period, it generates action potentials. But if you shine continuous light, it actually stops generating action potentials and silences its activity. We then tested the ability to silence the activity for treatment of arrhythmias. So to this end, we created a heart attack model in the rat heart of ventricular fibrillation. So we created a heart attack in the rat heart, took out the heart, and then, as you can see, it's beating happily. The scar is here. We are now pacing it in order to induce arrhythmia called ventricular fibrillation. You can see now the heart is fibrillating. And similar to the movies, you can say clear, but instead of giving an electrical shock, you give a light. And you can see that the lights restore the normal rhythm. Why is that? Because when we created the infarct, we also delivered the channel rhodopsin in the left ventricle. And when you gave prolonged light, it silenced the activity of the heart and restored eventually normal rhythm. So with that, I would like to end. I hope they show you some of the potential utilization we have for emerging biological techniques. And hopefully we can, using such approaches, bridge the gap between basic research and eventual clinical treatments. And with that, I would like to thank the many, many people in my lab, including Oren Caspi, that gave the introduction to our research, who did many of the work that I described. These are the many people that did our work. Most of them are actually MD PhD students from the Faculty of Medicine. I'd like to thank the people that we collaborate with over the world. And thank you for your attention.
Professor Gepstein, thank you for the great lecture and the overview of the field. So I encourage everyone to ask questions using the chat. And meanwhile, I'll ask a question. So focusing on the clinical translation, what do you think are the current major challenges for clinical use of human pluripotent stem cells, derived cardiac cells or cardiac tissues, or whether we should use cells or tissues in the clinic? And specifically, what do you think will be the appropriate indication for the initial application?
So you're asking some tough questions, of course. In other fields not involving the heart, there are already now clinical trials using pluripotent stem cells, either embryonic stem cells or iPS-derived cells. There are advanced clinical studies for macular degeneration in the retina. There are studies for transplantation of beta cells for diabetes, and they're planning to start studies for Parkinson's disease. In the heart, we have two major problems. First of all, we need a lot of cells. And second, we can really cause damage, because if you can cause a problem in the eye, the patient is already blind. You can cause damage, but not life-threatening, like if you cause, for example, a life-threatening arrhythmia in the heart. So in the heart, we are a little bit behind the other applications. The two major, I think, issues in the heart is, I think the major one is the concept of immune rejection. Do we give cells, allogeneic cells, that are expected to be rejected? And that means that we have to give some immune suppression. Can we give immune suppression in patients that we have not yet proven that we help them with the cells? So which type of patients to start with? Do we start with the most sick patients, in which maybe it's too late to help them? Or do we start with the people we have the most possibility to result in improvement, but we haven't proven yet that it works? So there's a question about patient selection. What do we do with immunogenicity? And in the large animal studies, I haven't talked about it, but there's evidence that maybe, at least initially after transplantation, these cells can cause arrhythmias, which can be dangerous. So one has to solve these problems before continuing into clinical studies. Nevertheless, I think for the other applications that I described to you, using these models to try to optimize the patient-specific therapy, choosing patient therapy based on the response of his cells, we are maybe advancing to maybe start guiding us for using iPS models to guide therapy, and also to as a model to develop new drugs that can eventually be used. So for this, I think we are closer to already using it in humans.
So something that relates to that, we have a question for Professor Karl Skoreski, who congratulates you for an outstanding, as always, lecture, and ask about the use of the iPS cell model as a platform for screening small molecules for electrical cardiotoxicity. So how close are we to translating that to the clinics?
So this is a very, very good indication, because I don't know if you know, but about 50% of all molecules that are being currently developed may cause electrical toxicity, similar to the patient that I described to you with the long QT syndrome. Interestingly, many molecules affect the same potassium channel that I showed you that had the mutation, but drugs can block it. And this is the #1 cause of withdrawal from the markets of drugs that were already approved, but eventually caused life-threatening arrhythmias in the patients. Much of this is because they were never studied on human heart cells before a clinical trial. So there's a lot of interest in the pharma industry to generate human-based heart models to screen the effect of drugs for cardiotoxicity. So we and others are trying to really establish models that will allow us to use the IPS models as a screening platform to assess for toxicity. The current challenges in what we're trying to do is first generating IPS from several different patients, from patients that are highly sensitive to drugs, so you can have a repertoire of cells with different genetic backgrounds. Second, in order to generate a reliable model, first of all, you have to make a purified cell-specific model. For example, to look at QT prolonging agents, you need to generate ventricular cells and not atrial cells. So I'll describe to you the advances we made in generating chamber-specific heart cells. Third, we need to make the cells more mature because our cells are relatively early stage. They are more like fetal tissue, while you want to see what drugs do to adult ventricular cells. And we also have a lot of advancements in the field in maturing the cells to become more like adults-like and to resemble the response of an adult cells. And lastly, we want to have high throughput screening methods in which you can test several compounds at different doses automatically. And we're working on this to try to scale it up. So you can study several, thousands of drugs at multiple doses, drug combinations, and so on, using automatic platforms and automatic phenotyping screening system. We are really optimistic that this will be the future of drug screening, at least at earlier stage of development, drug development.
So with that, if we don't have any further questions, I would just like to mention the next webinar on this great series of webinars for the Israeli Translational Research Association. The next lecture will be by Shai Soker about tissue organoid models for human development, disease and therapy development. Thank you very much, and thank you, Professor Gepstein, for the lecture, and thank you the organizers for the fabulous platform. Bye.
Thank you very much.